Gametogenesis is the production of sperm and eggs that takes place through the process of meiosis. During meiosis, two cell divisions separate the paired chromosomes in the nucleus. Then, the chromatids that were made during an earlier stage of the cell's life cycle are separated, resulting in gametes containing half the number of chromosomes as the parent. Hence, these cells are now in haploid state. The process of gametogenesis, where formation and maturation of ova or female gametes takes place, is called oogenesis. The female gamete is called an ovum or egg. It takes place in the ovary, which is the primary female sex organ present on either side of the lateral pelvic wall in the peritoneal cavity. The surface of the ovary is covered by a layer of cubical epithelial cells called germinal epithelium. The germ cells actually arise from the endoderm of the yolk sac and migrate to the genital ridge at about the 5th week of gestation and are called oogonia. They undergo several mitotic divisions by 6th month and reach to about 6 to 7 million oogonia in number. The egg in all animals is a larger cell in comparison to other type of cells. They contain reserve material such as yolk, glycogen, nucleic acids like DNA, RNA, etc. and various proteins. The size of eggs is small in smaller animals, while it increases in larger animals. Correspondingly, its number greatly reduces from small to large animals depending on their survival strategies. Sea urchin and frogs routinely produces hundreds or thousands of eggs at a time, while humans and most mammals produce only few eggs during their lifetime. The egg cell or ovum has three basic functions. 1. To supply nucleus containing half of the chromosomal component of the future embryo. 2. To supply almost all the cytoplasm to the zygote. And 3. To supply food reserves that will enable the embryo to develop up to a stage where it can begin to feed upon exogenous materials. Therefore, for these future needs, an egg cell becomes immotile, large-sized, well-specialized, packaged and programmed during a process known as oogenesis. By packaging, we mean the growth of the primary oocyte and the accumulation of condensed food reserves and substances within. The process of oogenesis is somewhat more complicated and different than spermatogenesis. Besides the production of 4 unequal sized haploid cells, there is acquisition of food reserves in the egg cytoplasm for the development of the embryo. Further, before the occurrence of meiosis, enormous amount of growth and differentiation of egg cytoplasm takes place. The oogenesis is more or less similar in all vertebrate groups. Oogenesis occurs in the outermost layers of the ovaries. The cells of the germinal epithelium detach from the surface epithelium and enter the cortex of the ovary. These germinal cells are diploid and are called primordial germ cells. Most of the primordial germ cells are approximately 10 micrometer in diameter. Oogenesis in insects. In insects, eggs are produced in the ovarioles of an ovary. The thinner part of an ovariole is called germinum, which contains oogonia developed by mesodermal cells. The stouter part of the ovariole is called the vitellarium, which contains oocytes in a longitudinal row. Each oocyte is surrounded by a single layer of cuboidal follicle cells. In the germinum, oogonia produces oocytes by mitosis. One oogonium divides into two cells, one primary oogonium and a cytoplast. The cytoplast divides by 4 consecutive mitotic division into 16 cells called cytocysts. One of these become an oocyte and the other 15 becomes nurse cells called trophocytes. All the 16 cells are surrounded by a single layer of flattened follicle cells. The oocyte and the nurse cells with their follicular covering come to the vitellarium, where the oocyte gets yolk granules from the hemolymph and the nurse cells. As deposition of yolk continues, the oocyte grows. Then by meiosis, the oocyte forms two cells, one big cell, the ovum, and the other very small cell, the first polar body. The latter degenerates after some time. In this way, the ovum gets half the number of chromosomes of the species. When the ovum is ready for the transfer to the oviduct, the follicle cells secrete the chorion or outer shell around it. In cockroach, the nurse cells are, however, absent. Oogenesis in mammals. In mammals, the process of oogenesis passes through 3 stages to form a fully formed egg, which are: number 1, phase of multiplication, number 2, phase of growth, number 3, phase of maturation. Phase of multiplication or proliferation. The primordial germ cells become the oogonia or the egg mother cell. The oogonial cells eventually undergo proliferation by repeated mitotic divisions, giving rise to the eggs and become primary oocytes when cell division ceases. From primordial germ cells to oogonia and up to the formation of primary oocyte, no conspicuous changes have been traced. Now, they enter into a period of growth. Phase of growth. Growth plays a much greater role in oogenesis than in spermatogenesis, as the egg contributes greater part of the substances used in the development process. The period of growth in the female gametes is very prolonged. The young oocyte of amphibians like frog may be about 50 micrometer and the mature amphibian egg is rather large and about 1000 to 2000 micrometer in diameter. In birds like hen, the diameter of ovum is as large as 40,000 micrometer, and in mammals it is only 200 micrometer. The rate of growth of oocytes also varies. It may be slow or fast. The period of growth in frog takes 3 years oogenesis. The first 2 years size increases gradually, and in the 3rd year accumulation of yolk increases its size. In human, it takes 12 to 13 years to grow. In hen, the last rapid growth of oocyte occurs in 10 to 14 days preceding ovulation, and during this time the volume of the oocyte increases 200-fold. In mammals, the proliferation of the oogonia is restricted to the intrauterine period of life. As the oocyte increases in size, the nutrients and other materials which have been synthesized accumulates in the cytoplasm. This progressive growth period and increase in nuclear as well as cytoplasmic substances of oocytes may be divided into 2 stages. Number 1, Previtellogenesis growth period, and number 2, Vitellogenesis growth period. Previtellogenesis growth period. During this period of growth, the nucleus and cytoplasm of primary oocyte increases tremendously in volume. Synthesis and accumulation of food reserve material called yolk does not take place. There is qualitative and quantitative increase in the amount of cytoplasm. The mitochondria increase in number. The network of endoplasmic reticulum with ribosomes become more complicated. The Golgi bodies manufacture cortical granules besides performing their normal function. Growth of nuclear substances. During this phase, due to the production of large amount of nuclear sap, the nucleus increase in size. In oocytes of animals having large eggs like amphibians, the chromosomes acquire a very characteristic appearance. A dark body appears at one place outside the nucleus, usually near the Golgi complex, and is known as yolk nucleus of Bulbiani. This large sized oocyte inflated with the fluid is now called germinal vesicle. The nucleus of the oocyte enters the prophase of meiotic division. Synapsis occurs between homologous chromosomes and each chromosome increase in its length. However, the amount of DNA in each chromosomes does not increase in proportion to the enlargement of the nucleus. The increased chromosomes look like bottle brush, hence they are called the lamp brush chromosomes. During the growth period of oocyte, all mRNA molecules are not utilized during translation, but some are inactivated by the wrapping of proteins around them and stored as informosomes to be used during early cleavage of egg, when chromosomal DNA remains more actively engaged in its own transcription of mRNA, rRNA and tRNA. The RNAs are transcribed by rDNA of nucleolar organizer region of chromosomes. The nucleus has a significant role in the storage and maturation of ribosomal RNAs. It also synthesizes all the proteins required for biogenesis of ribosomes. Therefore, during growth period of primary oocytes, the nucleus increases greatly in size and becomes very conspicuous. In many animals, particularly in amphibians, instead of one large nucleus, numerous small sized nucleoli are formed in their germinal vesicles. Most of them are localized on the periphery of the nucleus, immediately underneath the nuclear membrane. This high rate of gene activity is correlated with the fact that gene reduction, that is meiosis, does not take place until after the growth of the oocyte has been completed. As a result, the oocytes remain tetraploid for a long time. Growth of cytoplasmic substances. The amount of cytoplasm of oocyte increases both quantitatively and qualitatively during this period. Young oocytes in many animals show a very simple organization due to poor cytoplasmic inclusions and possesses none of the specialized structures found in the adult oocyte and mature egg. The cytoplasm is finely granular, having granules of ribonucleoprotein and DNA. Mitochondria are fairly scarce in young oocytes, but may increase in number during the growth of primary oocyte, because overall oxygen consumption increases during this time. In amphibians and birds, the mitochondria become aggregated in the form of large mitochondrial clouds. A growing oocyte has large amount of mitochondrial DNA than nuclear DNA. The young oocytes have the granular endoplasmic reticulum in the form of numerous small vesicles and annulated lamellae. These membranous structures appear in the form of stakes of cisternae, either in parallel or in spiral arrangement. Sometimes annulated lamellae are associated with ribosomes and RNA in high concentrations, and there is also an ATPase activity in the pore complexes of these lamellae. The lamellae thus serves as a storage site of RNA in cytoplasm, and they are found to break down and disappear during late oogenesis. In young oocyte, the Golgi bodies are found around the centrosome. In mature oocytes, they form a large spherical mass in some mammals or become located in the subcortical cytoplasm of frog and chick, or sometimes may disappear completely. The Golgi complex of oocyte is believed to synthesize cortical granules besides performing its normal function. In the cortical region, cortical granules are present. These are membrane-bound spherical bodies of diameter from 0.8 microns in sea urchin to 2 microns in frog and contain acid mucopolysaccharides. These mucopolysaccharides are used during fertilization in the formation of fertilization membrane. They are present in bivalve mollusks, some annelids, fishes, frogs and some mammals like rabbit and man, but are absent in some insects, gastropod eurydiales, birds and some mammals like rat and guinea pig. These granules are synthesized by cisternae of Golgi complex in the interior of the oocyte, and later they move to the periphery where they are arranged in a layer close to the plasma membrane of oocyte. Vitellogenesis growth period. The process of formation and deposition of yolk is called Vitellogenesis. In amphibians and fishes, its synthesis takes place inside the modified mitochondria. In some insects, yolk formation occurs in fat bodies, whereas in most of the vertebrate, yolk is formed in the liver of the mother. From the liver, it is carried by blood to the ovary. The yolk percolates to the follicle cells and from there to the cytoplasm of the oocyte. However, in vertebrates, a very small quantity, about 1% of the yolk material, is synthesized by the oocyte cytoplasm itself. Thus, a major part of the material forming the yolk is exogenous. Yolk is a general term that covers the major storage material such as glycogen, certain other carbohydrates, proteins and lipids. Hence, Vitellogenesis is the period of rapid growth. The growing oocytes are surrounded by spatial nutritive cells which help in their growth like follicles and nurse cells. They supply nutrients to the oocyte during growth phase. Phase of maturation. The maturation phase is accompanied by two maturation or meiotic divisions that differ greatly from the maturation division of the spermatocyte. Here, after the meiotic division of the nucleus, the cytoplasm of the oocyte divides unequally to form a single large-sized haploid egg and three small haploid polar bodies or polocytes at the end. This type of unequal division has great significance, which allows one cell out of the four daughter cells to contain most of the cytoplasm and reserve food material sufficient for the developing embryo. First maturation division. During the first maturation division or first meiosis, the homologous chromosomes of the primary oocyte nucleus passes through pairing or synapsis, duplication, chiasma formation and crossing over. A new nuclear envelope is developed around the daughter chromosomes by the endoplasmic reticulum. After the karyokinesis, unequal cytokinesis occurs, forming a small haploid polar body or polocyte and a large haploid secondary oocyte or oocyte. When the primary oocyte finally completes its first meiotic division inside the primary follicle, it divides the chromosomes evenly. However, it does not divide its cytoplasm equally. Most of the cytoplasm remains in one of the two daughter cells, which becomes a secondary oocyte. The other daughter cell, which gets half of the chromosome but very little cytoplasm, is called a polar body or first polocyte. The polar body is not a functional oocyte. Instead, it degenerates and dies. The formation of a polar body allows the primary oocyte to reduce its genome by half and conserve most of its cytoplasm in the secondary oocyte. Second meiotic division. The haploid secondary oocyte and first polocyte pass through the second meiotic division and forms mature egg and a second polocyte. By the second meiotic division, the first polocyte also divides into two secondary polocytes. These polocytes ooze out from the egg and degenerate, while the haploid egg cell becomes ready for fertilization. By such uneven division, half of the chromosomes are retained by the ovum along with almost all of the cytoplasm. The polar bodies eventually go through atresis and degrade. During the process of oogenesis, oocytes of many animal species undergo meiotic arrest prior to completion of chromosomal reduction, and it is in this stage they undergo tremendous growth. The length of time that oocytes remain in this arrested state and the nature of the stimulus which reinitiates meiosis are species dependent. Hormonal control of oogenesis. Gonadotropin release hormone secreted by hypothalamus stimulates the anterior lobe of pituitary gland to secrete luteinizing hormone or LH and follicle stimulating hormone or FSH. FSH stimulates the growth of Graafian follicles and also the development of egg or oocyte within the follicle to complete meiosis I and form secondary oocyte. FSH also stimulates the formation of estrogens. LH induces the rupture of mature Graafian follicle and thereby the release of secondary oocyte, a process called ovulation. The remaining part of the Graafian follicle is stimulated by LH to develop into corpus luteum, which secretes progesterone. The rising level of progesterone inhibits the release of gonadotropin release hormone, which in turn inhibits production of FSH, LH and progesterone. FSH and LH act on a primary follicle in the ovaries, promoting its development into a secondary follicle. FSH and LH promote meiotic maturation division of oocyte and stimulate follicle cells to synthesize vitellogenin. Theca cells are recruited to the oocyte and estrogen production rises. The estrogen level is controlled by negative feedback and acts on the hypothalamus and pituitary gland to switch off gonadotropin release hormone, follicle stimulating hormone and luteinizing hormone secretion. One oogonium produces one ovum and 3 polar bodies. Polar bodies have small amount of cytoplasm. It helps to retain sufficient amount of cytoplasm in the ovum, which is essential for the development of early embryo. Formation of polar bodies maintains half the number of chromosomes in the ovum. During meiosis, first crossing over takes place, which brings about variation. Oogenesis occurs in various organisms. Therefore, it supports the evidence of basic relationship of the organisms. Without oogenesis, there is no fertilization, reproduction and life. The process of gametogenesis, where formation and maturation of ova or female gametes takes place, is called oogenesis. The female gamete is called an ovum. Oogenesis involves the progressive elaboration of oocyte and includes transport of yolk proteins into the oocyte, the localization of mRNAs, and the production and translocation of cortical granules, which lie immediately beneath the plasma membrane and are crucial for events following fertilization. Most of the biochemical changes in the oocyte takes place in the growth phase. Oogenesis varies among species to species. It involves not only the production of haploid nucleus, but also the acquisition of food reserves and primary organization of the cytoplasm, from which embryo will be formed.